Well, wherever you're joining us, a very, very warm welcome to today's MycoTalks. Today we have two fantastic speakers, Jenny Lodge from Duke, and then we're having, after that, Amelia Barber from Hans Null Institute in Germany, as you can see there. If I can have the next slide. I just wanted to give you a bit of other information. MycoTalks is here to provide a wide diversity of talks covering all aspects of medical mycology, and it includes people of all career stages, gender, geography, and area of research. And what we do, as those of you who have joined us in the past will know, we have two 30-minute talks, no questions in between, and then we have a 30-minute in-depth discussion. Now, for that, please do put at any time during the two talks your questions in the Q&A part of the screen. Don't use the chat, please use Q&A. And Joe Heitman, my co-chair, and I will screen those and be able to ask those to our two guests today. So if I could have the next slide, I just wanted to also signal to you that we've got on November the 30th, Sandy Johnson and Matt Fisher. So that would be a fantastic session as well. And then on 25th of January, Damien Kreisen and June Round. In addition to the MycoTalks, if we have the next slide, we also have our medical mycology trainee seminar series, and that's for students, postdocs, and clinical trainees and new faculty. So it's a great chance for you to present your work to a global community, and those are on the second Thursday of the month, but we can make some adjustments for that. So if you want to be involved in that, you'll see there's a place that you could submit an abstract. So do join in with that. It's a great, great session as well. And then finally, the last slide. Yep, we also have quarterly our myco clinics, which is focused on more clinical aspects of medical mycology, presenting things like case highlights and the challenges of all things to do with clinical mycology. So we have a number of opportunities for you. And just note there, on December the 7th, there'll be one on chronic aspergillosis. So please join in with that. Okay, now let's get going. So the first of our two speakers today is a great colleague and friend of mine. So I'm really delighted to welcome you, Jenny Lodge, who's a professor of molecular genetics at microbiology at Duke. And she has a senior position there as Vice President for Research and Innovation. And that's a very prestigious post handling a very large research portfolio at Duke. She came to Duke in January 2022, but many of us have known her for much longer than that. Previously, of course, she was at the School of Medicine at Washington University in St. Louis. And you will know, or at least many of you will certainly be aware of her really excellent research over many years on the human pathogenic fungus Cryptococcus neoformans. And she's been funded to do that work for many years, and she's done a lot of different types of work, but particularly had a great focus on how the fungus builds its cell wall and the immunological consequences of various things in the wall. And she's been recognized with many awards for her work over many years. So at that, I'm going to sign off and welcome you again, Jenny, and let you take forward the first of our two talks today. Welcome.
Thank you so much, Neil. It's really a delight to be here and an honor to be giving one of the MycoTalks. I do know that, and I'm going to talk about Cryptococcus and the cell wall and the immune response of the cell wall and how the cell wall changes. And I know that last month was Carol Monroe, and she gave a fantastic talk on the cell wall of Candida albicans. So I hope this builds on this, and that some of these, what I'm going to talk about today, is isn't too unfamiliar to you. But Cryptococcus is a fungal pathogen. It is found ubiquitously around the world. It grows often as a yeast form, where it's basically looks like a budding yeast. It is found in the environment in many different places in the soil, associated with plants, associated with bird excreta, and it can also create hyphae and produce basidia that then produces long chains of spores. These spores can then be inhaled. And in a normal, healthy host, they get into the lung, but there's the host response is such that it either eliminates the infection, or it can wall the Cryptococcus off in some way, and basically prevent it from causing disease. However, if the person is immunocompromised, and particularly with HIV during late stage AIDS, this infection can either reactivate, or if it's a new infection, it can disseminate to the central nervous system. And this cryptococcal meningitis is invariably fatal unless treated. And so it has created a lot of problems, particularly in our HIV AIDS population. It has tracked pretty well with AIDS and in Western countries, and now it's actually very prevalent in sub-Saharan Africa, where HIV is still a major problem. Part of the issues with Cryptococcus is that often it's diagnosed late. There's not very many antifungals that are effective and non-toxic. And so there's still a need to develop more antifungals. There's no vaccine, but I'll talk a little bit about that. There's a lot of people who are interested in this. And it's still causing just under 200,000 deaths per year, it's been estimated. And most of these are AIDS patients, and in AIDS patients, it causes about 15% of the deaths that are associated with AIDS. I'm going to talk a lot about the cell wall of Cryptococcus. Cell wall is obviously important for structure and morphology, protection from the environment. It's also the face of the fungus that interacts with organisms outside, other organisms, whether that be other microbes or the hosts, mammalian hosts. And it also provides attachment of virulence factors. And this particular photograph here is a beautiful electron micrograph that was taken by John Heuser many, many years ago. And it's a freeze fracture deep etch scanning micrograph. And what it shows is this is the membrane of the cell, and then it's been fractured off. And you can very clearly see this green here is the wall. And you can clearly see two layers. One is sort of very densely packed and looks very organized. The outer wall seems to be less densely packed. And then the purple here is the capsule that comes out of the wall. And the capsule is a major virulence factor for Cryptococcus, as is melanin. And you don't see it here, but melanin is thought, because these cells weren't grown in the presence of the appropriate substrates, but melanin is thought to actually sit in the inner layer of the wall. And one of the things that is so attractive about the wall, and why so many people are interested in it, is that it's very unique to fungi. And so humans don't have a lot of the components or the counterparts of the enzymes that are involved in biosynthesizing the wall. And so it's a great target for antifungals. One of the caveats there is that it also is quite dynamic in that the wall can tolerate indicate a lot of differences in the amount of the various components that are produced. So for example, if you make a little less beta glucan, you might be able to compensate other ways. It is comprised mostly of an intermesh of carbohydrates and as well as various proteins, mostly nanoproteins. Oops, sorry. How do I get back? The Saccharomyces has a much simpler wall in comparison to Cryptococcus. Saccharomyces has beta-1,3 glucans and beta-1,6 glucans and chitin and these manoproteins. And many of these manoproteins started out as GPI anchored proteins. They start in the membrane, and then the GPI anchor is clipped off and it moves to the wall. And is shown to be attached to beta-1,6 glucans. And we've got a little data that says that that's not happening, that we actually see that movement of the manoproteins from the being GPI anchored in the membrane and going out to the wall and having the GPI anchor clipped off, but it's not covalently attached to beta-1,6 glucans. Cryptococcus also has melanin. It converts a good part of its chitin and deacetylates it to chitosan. It has alpha glucans, which are important for the capsule attachment. And it also has this capsule. So the composition and the organization of the crypto wall is quite complex. Actually, it has quite a lot of chitin compared to some other fungi, and it converts a lot of that to chitosan. And one of the things that's interesting is that if you look down here, Candida and Saccharomyces, if you look at chitin during growth, it's fairly level, whereas in Cryptococcus, as the cultures grow denser, it actually produces more chitin, and you can see it's actually turning much of that chitin into chitosan. So this is a dynamic process that seems to depend on the growth phase. And there are other fungi that also produce chitosan. Mucor, the black molds are a particular example that makes lots and lots of chitosan as well. Aspergillus makes a little bit, but Candida and Saccharomyces don't make any. Saccharomyces makes it during sporulation, but that's really it. And so chitin is a polymer. The building block is UDP glycnac, and chitin synthases are membrane-bound proteins with lots of transmembrane domains that sit in the membrane and produce chitin. There's lots of different classes of chitin synthases, and some of those chitin synthase require an accessory protein. And in Cryptococcus, we've called these CSRs for chitin synthase regulators. And the CSRs are prenylated proteins that also sit in the membrane. Chitin is fairly insoluble and neutral and rigid. And Cryptococcus has 8 chitin synthases, which has made it a little more challenging to study some of the specific activities of each of the chitin synthases. I know out of Neil's lab, they've done some really beautiful work looking at different types of different structures of chitin that is produced in Candida. And we haven't been able to do that kind of work in Cryptococcus, so we're quite jealous. Chitosan is the deacetylated form of chitin, and it's not necessarily that you have a strand of chitin that's 100% acetylated, and then you 100% deacetylate that, and now you have chitosan. There's all this range of deacetylation, percent of deacetylation, and patterns of deacetylation that can occur, and that's something that we're just starting to explore. Chitosan is it's more soluble than chitin. It's flexible, and so it really has very different physical properties than chitin does. And in Cryptococcus, there are 4 deacetylases. 3 of these have GPI anchors, and the other one does not. So chitin deacetylase 1, 2, and 3 all have GPI anchors, and CDA4 does not. They can all be found in the cell wall. The ones with GPI anchors can also be found in the membrane when the GPI anchor is still intact. And many of these were identified early on as immunoreactive proteins. These are heavily monosylated proteins, and they're also, once they get out into the wall, they can also be secreted, and so they can be found in the media. And so we have a working model of how chitin and chitosan is produced. Well, particularly the chitosan, and we've got genetic data, but I'll just sort of give you the overview of the model, and that is that we have got one specific chitin synthase out of the 8, and this is chitin synthase 3, that is sitting in the membrane. It requires an accessory protein, so this CSR2 also sits in the membrane with its prenyl tail here, and then there are chitin deacetylases that are GPI anchored, that are also in the membrane, and that they are deacetylating the chitin as it is being produced. Other chitin synthases would be just producing plain, just regular chitin without deacetylation. And that we'll also believe that at this point that there's the 4th chitin deacetylase, CDA4, which we initially called FPD1, is out in the wall and is actually deacetylating some of the chitin out in the wall. And the evidence we have for this is that when we delete, the genetic evidence at least, is when we delete CHS3, the chitin synthase, we basically get no chitosan. There's none or very little produced. You can see it over here. Here's the CHS3 deletion. The red is the chitosan. Here's the wild type, lots of chitosan, very little chitosan. And so the reason we believe that the CSR2 is its partner is because when we delete CSR2, we see a very similar phenotype of very little chitosan compared to wild type, and it's very similar to the CHS3. And then when we delete, well, I don't have a view for this, but when you have either CDA1, CDA2, or CDA3, in the cell, then you produce chitosan. But if you delete all 3 of these, so there's some functional redundancy here. If you delete all 3 of them, now you produce 0 chitosan. And so you can see that there's no red in these cells. And you've got to keep in mind that there is still CDA4, which is out in the wall, not GPI anchored. And so we've looked at the localization. This is work done by Nicole Gilbert. And the CDA2, when she had a very nice antibody, and she could see that it was both in the membrane, so it was attached here, but you could also find it in the wall. It was also secreted. And it was not the protein that was in the wall, the CDA2 protein that was in the wall was not covalently bound. You could wash it out with SDS or with sodium chloride. It didn't require, we had strains that didn't make beta-1,6-glucan, and it was still in the wall, so it didn't require a covalent attachment, and it wasn't released by HF pyridine, which is used to release those covalently attached GPI anchors. So I think this begged the question of where is this CDA2 active? Is it active when it's in this complex, or is it active when it's out in the wall? And so we were able to remove the GPI anchor signal from the CDA2 protein, and basically when we do the localization, there's nothing in the membrane, but it's all in the wall, but no chitosan is produced. Then when we can make changes to the GPI anchor signal and restrict it to the membrane. So when we do the localization, there's nothing in the wall fraction, and it's all in the membrane, and we see chitosan produced at that point. So we believe that these CDAs, this at least chitin synthase 2, or chitin deacetylase 2, is only active when it is intercalated into the membrane in a complex with chitin synthase 3 and chitin synthase and CSR2. And we're busy trying to tag all of these proteins in order to do really robust pull down assays. We've got some old data that suggests that they are in a complex, but we'd like to clean that up. So then the question is what's CDA4 doing? There's no GPI anchor. It's found in the wall and secreted. It's not found in the membrane. Bruno Morschbacher's lab published a paper a couple of years ago showing that the substrate specificity for CDA4 actually required a partially deacetylated substrate. So we think that if you are just making chitin out of a chitin synthase without any deacetylation, you can't get any chitosan. It's not going to produce, it's not going to be able to use this 100% chitin as its substrate, whereas if you've got this complex going and you're partially deacetylating the chitin chitosan strand, it can actually go in and continue to deacetylate. And so we're very excited about that. And Maggie Hellman, who's out of Bruno's lab, was in our lab for a couple of months this spring. And she's really interested in what the patterns of deacetylation are that's produced by the different chitin deacetylases. And so she's back in Germany and is busy doing NMR analysis of a whole bunch of the strains. And so we can't wait to see what Maggie has come up with. And so if we go back to these chitosan deficient strains, so we've got three. One is the triple CDA123 mutant. We've got CHS3, which doesn't make much chitosan. We've got CSR2, which also doesn't make much chitosan. These strains were all very rapidly cleared. If we give it sort of a normal inoculum that causes disease generally in about 20 days, it causes lethality in about 20 days if you inoculate with 10 to the 5th organisms. But for those were very rapidly cleared within 48 hours, all of the chitosan deficient strains. We knew that the rate of clearance is also dependent on inoculum size. So if you give it 10 to the 7th, now we actually see after a day or two, there's still plenty of even the chitosan deficient strains. This is the CDA123, the triple mutant. But you can see that over time, by day 7, it's clearly in the process of being cleared. So it just sort of changes the time frame for how fast things are cleared. But I'm showing you this because we're going to be using 10 to the 7th as an inoculum for other things coming up. The other thing that we've shown is that these are all completely avirulent in a mouse model. They're cleared, they don't cause disease in the mouse model. And so we were interested in what the host response was, and not being an immunologist, we are lucky enough to be working with Stu Levitz and Charlie Specht up in UMass, and also Cameron Hole and Brian Maybrook were postdocs and senior scientists who came into the lab that had immunology expertise. And so that's been great. And so what we've been looking at is, what are the cytokines and chemokines that are induced by these different strains. So now we've got our chitosan deficient triple mutant and wild type over here. And what you can see is that there's a massive Th1 response, which is going to drive inflammation when we inoculate with this chitosan deficient strain. And so then the question was, could this possibly, if we're changing the host response, would that actually be, could we actually get protection? Could this be a potential vaccine? And so what Raj and we did were they inoculated mice with a high dose of the chitosan deficient strain, waited for 40 days, and then challenged with wild type. And basically what we see is when we inoculate, you get the mice continue on happy and healthy, whereas if you don't vaccinate, then they die as normal in about 20 days. And so that was very exciting. A lot of people are working on vaccines for fungi, and especially for Cryptococcus, and people are using all kinds of things like purified antigens, anti-capsular antibodies, glucan particles that have purified proteins in them, activated host cells. And then there's a lot of people who are doing engineered or mutant crypto strains. And I think this has been a real boon over the last, 8-10 years that people have started discovering all kinds of strains that are can be acting as vaccines. And so that's been a very exciting development, especially as we start to try to understand what's similar and different about these different strains. And one of the concerns that people always have is that trying to use a live attenuated strain as a vaccine. And so what we did is we looked at heat-killed cells and found that those also worked quite well as a vaccine. We started thinking, what's the difference between our chitosan deficient strain versus wild type, because wild type in general does not act as a vaccine, but I'll give you a little, that's not quite true. It depends. But one of the questions we had is, is it chitin? Is it more chitin that's present in the cell, or is it the lack of chitosan that's driving these responses? Maybe the cell wall is just very disordered and other epitopes are exposed. Or maybe it's because we don't have that CDA protein. Remember, those are immunoreactive. And so maybe those were important to drive the protective response. And so we don't know completely the answer at this point, but we're still working on that. This just shows that in wild type, you can see lots of chitosan that's stained specifically with ESNY and not a lot of chitin on the surface with wheat germ agglutinin staining. Whereas with the triple mutant, we definitely see CDA. We definitely see the chitin being exposed on the cell surface and no chitosan. And this is similar to results that have come out of Andy Allspa's lab as well. We've looked at the wall structure, and this is once again a freeze fracture deep etch microscopy. And so this is the wild type, and you can very clearly see this is the wall, and you can very clearly see the two layers, the more highly dense ordered layer, and then a sort of a looser layer up here. And in our chitosan deficient strain, that structure seems to be gone, and that this is all sort of a very highly disordered layer. So it's quite possible we are seeing other epitopes being exposed. And then we also asked the question about whether or not it's the chitin acetylase protein itself that is that is so important. And what Raj did is he made a lovely mutant that was catalytically inactive. It was highly expressed, and basically even the presence of that protein doesn't change the host response or the protection. And so we don't believe it's the lack of the CDA protein that is driving the response, but I think these aren't mutually exclusive. It could be a combination of the other ideas. Charlie and Stu have been working hard at trying to understand what cells are required to induce or maintain protection. And so they've looked at various mouse strains and that are lacking in particular cells. And this is only a partial list of all the things they've been working on. They have a huge list of things they're doing. But basically, if you get rid of all B cells and all T cells, you don't get any protection. So obviously, either B cells or T cells are required. If you have a mouse strain that doesn't make CD4 T cells, you don't get protection. And that's, of course, concerning because we're targeting an AIDS population, but these completely lack CD4 cells. And then there's a strain of mice that lacks B cells, and we do see, and Charlie and Stu do see protection in that strain. And so it looks like the B cells are not important for induction of this protection. And so to get at the question of which CD4 or CD8 T cells might be required, we did a series of T cell depletions using anti-CD4 or anti-CD8 antibodies. Basically what we see is if we, and this is work that was done in our lab as well as in Charlie and Stu's lab, where we're trying to repeat our key experiments so that we're sure that they're all reproducible and accurate. And what we see is that if you deplete CD4 T cells before you vaccinate, then you don't see any protection. And so that means that the CD4 T cells are important for induction of the protective response. But if you, in this graph shows an experiment where we depleted CD4 T cells after we vaccinated, and then we still see protection in that situation. So it suggests that the CD4 T cells are important for induction of protection, so that if you vaccinate while somebody still has their CD4 T cells, they will be protected, because the CD4 T cells, when you're in late stage AIDS and those cell types are dropping, that the protection will still be there because they're not important for the maintenance of the protection. One of the other things that we observed, and this is Cameron Hole, who is a postdoc in the lab, and he was looking at CHS3 to see whether or not that might be a vaccine strain as well. And to his surprise, when he inoculated with a very large quantity, the mouse died right away, like within a couple of days. It was really quite surprising how quickly the mice died. It turns out that the CHS3 strain was inducing massive inflammation and a massive influx of neutrophils, and that he could deplete neutrophils and show that the neutrophils is what was causing the mortality. And he did heat-killed cells as well, so it's not about the CHS3 creating an infection. It really is the host response. And so what we've got now is we've got this sort of hyperinflammatory response that causes death. We have sort of a moderate inflammatory response where we see protection, and that's the CDA123 triple mutant, where we see a Th1 response, but we're also getting protection. And then with the wild type, we got very little inflammatory response and no protection when we grew cells sort of under our normal conditions. And so one of the things that we asked was, is it possible to have a little, just can we reduce this inflammatory response? Can we separate that from the protective response? And one of the things that Raj had observed is that he was seeing different chitosan levels depending on how we grew our wild type cells. So this is, the rest of this work is all in wild type cells. And so basically when you grow in unbuffered yeast nitrogen base, the pH drops dramatically from about 5 to about 2 within about 48 hours. And then the yeast nitrogen base at pH 7 stays fairly constant, and you can see there's less there. And so one of the things that we observe then is that When we grow in yeast nitrogen base unbuffered, we see a lot of epitopes exposed. So we see lots of calcofluor white staining. We see lots of WGA, which binds the chitin staining. We see more staining of the beta glucans, and we see more staining with con A as well. When we look at these unbuffered cells under the microscope, this is transmission EM, you can see that their walls look quite aberrant, and it looks like they're kind of peeling off a bit. This looks like a mechanical break, whereas the wild type cells grown in YPD or yeast nitrogen base look fairly normal. As Joe Vaselli, who's a new postdoc in the lab, has been looking at these pictures, and one of the things that he's observed is that it looks like we've got cells that are dividing and pulling apart, and they're actually ripping the outer wall off of the pair. So you can see that here, and you can see that here, and that's so we think that what's happening is that these cells are basically ripping the part of the wall that is sort of protecting it from the immune response off. And what we see then is we also see a massive Th1 response for those. And then interestingly, we were looking at the yeast nitrogen, the cells grown in yeast nitrogen-based pH7, and this had very little immune response or Th1 response, so even less than cells grown in YPD. We looked at the high doses of the cells that were grown in yeast nitrogen base unbuffered, and we basically saw that rapid death that was about an immune response. And when we tested the yeast nitrogen base pH7 grown cells, so these are the ones that had a very low Th1 response, or if any, and what we see is that this actually works as a vaccine. And this is work of Raj and we. So we're really excited, and I am about to wrap this up. And it is possible to have just a very small amount of inflammation or no inflammation, but have protection. And so we're really excited that we've been able to separate those two, and are looking forward to understanding what those different epitopes are that might be driving those different responses. I think the important thing here is that that the CD4 cells are important for induction of this protection, but may not be essential for the continuation, and that we may be able to induce a protective host response without inflammation. I'll just wrap it up by thanking everybody in the lab. Here's part of the current lab, as well as Anna and Eddie, who are our newest members. Anna's looking at echinocandins and where they are located, what they bind to in Cryptococcus. I've talked about all of these folks who are former lab members, as well as our collaborators from UMass and my longtime collaborator, Maureen Donlan, at St. Louis University. So I will stop there.
Terrific. Thanks very much, Jenny. Well, it's really a great pleasure to introduce our next MycoTalks speaker today, Amelia Barber, who is an independent junior group leader at the Friedrich Schiller University in Jena, Germany. And I had the opportunity to visit Jena earlier this year, and it was really terrific to witness the vibrancy of Amelia's research program in person. The focus of her research combines experimental and computational approaches to discover mechanisms of virulence and drug resistance in human fungal pathogens with a One Health perspective. And the title of Amelia's talk today is From Environment to Infection, Insights from Aspergillus fumigatus. Take it away, Amelia.
Great. Well, thank you so much, Joe, for the invitation, as well as the opportunity to speak in this really fantastic seminar series. And let me just share. Okay, well, hopefully you are seeing my slides correctly. And today I'm excited to tell you, like Joe said, more about Aspergillus fumigatus, and particularly a bit about its life in the environment, which people often don't think about, including what sort of fungicides it is exposed to and the consequences of this, as well as dive into what we've learned about its virulence and success as a pathogen using some next generation kind of new systems biology approaches. And so Aspergillus fumigatus, in comparison to other fungi that are commensals of us, this is a fungus that actually primarily lives in the environment. And in the environment, it's a saprobe, which means it degrades dead or dying plant matter. And its primary substrates in the environment are things like soil and compost. And in the environment, it can grow as a mycelium, but then it's also really good at producing these spores, which are the infectious particles for us as humans. And so if, the way we get exposed to Aspergillus is when we inhale these spores, and the infections tend to be pulmonary in nature. And these spores, they are very tiny. They're very good at going very deep into the lungs and creating a very invasive pneumonia if you're immunosuppressed, or we're increasingly realizing if you have damage to your lungs from something like severe COVID. And the case fatality rate for these infections is very high, and then is incredibly high if it's for a resistant infection. And so many of us in the Aspergillus community, we always say, or at least I always say, we're exposed to this fungus kind of no matter where we are in the world, and probably almost daily. And we've never really had a number to back this up. And the statement is based on that, when clinically the disease presents, across all the continents, as well as, if people, when they've done targeted sampling, you can usually find it in most places. And so now I just wanted to show a little bit of new data from our lab, really highlighting how ubiquitous this fungus truly is. And so this is some data that was generated by Shinyu, who is a really motivated master's student in the group and is very interested in kind of what sorts of fungi we are exposed to in our daily lives. And so to answer this question, she's been taking advantage of the large amount of publicly available sequence data, including in soil and air, and mining them for what sorts of human fungal pathogens are there. And so this is the data for Aspergillus fumigatus. And we have our sequencing projects here. We have 221 that we identified a good fungal signal for. And we can see in basically about half of these soil microbiomes, at least one of the samples is positive for Aspergillus fumigatus. And then if you look at the value for the entire 7,500 samples, 1 in 4 of them is positive for Aspergillus fumigatus. So this is a fungus that is truly globally ubiquitous. And unfortunately, one of the other challenges about Aspergillus fumigatus is that it's the poster child for increasing antifungal resistance. And I think this is nicely highlighted on this map here, which is not just about Aspergillus fumigatus, but really underscores the problem of antifungal resistance, not only in human pathogenic fungi, but also plant pathogenic fungi. And this increasing antifungal resistance is really bad news because, while there are more than 16 classes of antibiotics available for treating bacterial infection, we only have 3 main classes for treating human fungal diseases. And for treating aspergillosis, management of these infections really relies on one class, the azoles. And incidentally, the azoles are actually, they're used not only for plant protection, but also for human medicine. And their use as a fungicide predates their use as a human antifungal. And so this is just some data that I put together looking at the number of approved triazole compounds for both agricultural and clinical use. And we can see that numerically, there's actually 5 times more azoles approved for plant protection. In this context, they play a very important context because, we as a human society, we need to have enough food to eat. But the bottom line is, regardless of whether the compound is used for agriculture or clinical use, they really are thought to have the same mechanism of action and target the same fungal enzyme. And so, like several other fungi, we have this question in Aspergillus of really where is this increasing antifungal resistance coming from going from basically non-existent 30 years ago to now the global resistance rate of Aspergillus is about 8%, and there's areas of the world where it exceeds 20%. And so given this dual use context, it's been hypothesized that actually it's azole use in the environment on things like crops, where a huge volume is applied, is driving resistance. And additional support from this comes from the fact that really the clinical use of azoles is not really thought to contribute because Aspergillus does not spread patient to patient because the fungus doesn't produce conidia within the host typically. And so to really test this hypothesis and look at the link between azole use on crops and the development of resistance, we did a One Health study in Germany when we performed soil sampling on crop fields before and after azole fungicide application. And this was done in the collaboration with the University of Halle in Germany. And at this point, I'd also like to acknowledge Jenny, who you can see here on the screen in a wheat field, who did a lot of the soil sampling for this project. And you can also see from the photos that most of our field sites are on cereal crops, so things like wheat and barley. But we also have a couple apple orchards that we've been sampling on. And we did this sampling over 3 consecutive growing seasons. And so the first question that we really wanted to answer is, how much Aspergillus fumigatus is in soil and how does it change upon fungicide exposure? And so here we're going to be measuring that as the number of CFU per gram of soil. And we can see that if we compare the levels that are in the soil or on these different farm fields in April, which is before the vegetative period and before any sort of fungicides are applied, and compare it to the levels after fungicide is applied and after the growing season, we can see that generally we see less Aspergillus fumigatus there. However, I will also say that we did this over multiple different years, and the trend is a bit more variable. So I think the final conclusion I would feel comfortable saying is that, I think there's many factors that impact how much Aspergillus fumigatus is in, say, soil, and fungicide application might be one of them, but it's certainly not the only thing. But really, one of the main motivations for doing this study was to see what sort of changes in antifungal susceptibility accompany real-world fungicide exposure. And so here we're going to be measuring that as the minimum inhibitory concentration, or MIC. And we're going to start by looking at the distribution of MIC values to 2 very heavily applied agricultural azoles, difenoconazole and tebuconazole. And what we can see here is if we compare the MIC distribution in these strains before fungicide to after, you can see that we do see this modest shift upwards in the MIC curve for both drugs. But you'll also see that actually the median values don't change, saying that, even though some strains change, it's not a lot, and most strains do not change. And this is something we saw for our agricultural fungicides here, as well as our 2 medical azoles, itraconazole and voriconazole. And for these drugs, we also have a clinical breakpoint. And this is where we say, if this strain came to the clinic in a patient, we would label it as resistant because therapy would start to fail and we would start or struggle to manage these infections with an MIC above this value. And so when we look at our medical azoles, we can see the same general thing where we do see a bit of an upward shift in the MIC curve. But again, most things don't change, as well as when we look at the amount of isolates that would be considered actually clinically resistant, it's very, very low. And so we can actually quantify this over the 3 years that we did our sampling and see that from this large collection of strains, when we look at them, only 1 to 3% of agricultural Aspergillus fumigatus isolates that were pulled, directly from soil that were treated with fungicides would be considered clinically resistant. And so the last thing we wanted to look at with this data set and in the study was really, what is the genomic impact of fungicides on these? Maybe we don't see a big change in antifungal susceptibility, but we can still find evidence of a population bottleneck or something like that. But I will save you the time and say we did not really find any significant impact. And I'll just show one piece of data here that's called Analysis of Molecular Variation, or AMOVA. And it basically says when we did whole genome sequencing, for all of the strains that we sequenced, say, on farm field B, where does the variation in this population come from? And you can see that for all 4 of the strains that we had before and after isolates from, the majority of the variation is strain level variation. And then the amount of genetic variation that can explain whether an isolate is from before or after fungicide exposure is this tiny gray bar up here that you probably can't see and is not statistically significant. And that's the amount of variation that's explained by whether an isolate is from before or after fungicide exposure. And so our conclusion from this One Health study is that really, contrary to what we thought and what has been hypothesized, we don't find evidence that fungicide use on crops is driving this. And we base this on the fact that, when we looked at the MICs of isolates, often the level that was clinically resistant was very, very low, as well as when we looked at the genome level, we couldn't find any impact. But it still leaves this open question of really, if it's not coming from use on crops, which I would add is a good thing, because this means that, we as a human society do need food to eat. So there might be some contexts where it's okay to keep using this and kind of maximize crop yield. But we do have this question of where, if not here, where is this resistance coming from? And so here we'd like to posit this is the work of myself as well as others here, that really it's that not all contexts contribute equally to the resistance development for Aspergillus fumigatus, particularly with regards to the azoles. And we think, there are certain cold spots that don't really contribute. And this is things, like agricultural soil, like we showed in our study, as well as some others have done work on harvested cereals and silage and found that these don't really seem to contribute significantly. On the other hand, we have hotspots, and these are things that probably are contributing. And these are things like flower bulb waste, because ornamental plants are typically treated with a much higher concentration of fungicide than anything that we need to eat, as well as things like green waste and compost, where as the plant material breaks down, you actually can get an increased concentration of the fungicide, and this can have a higher selective pressure. And so moving forward, I think, my group and as well others are really saying that it's these hotspots that we really need to work on mitigating to prevent increasing resistance of Aspergillus fumigatus. Okay, so now I want to bring it back to something at the beginning that I said, which is that, the environment is the primary habitat for Aspergillus fumigatus. And this is actually, it's not special to Aspergillus fumigatus at all. In fact, 3/4 of the WHO priority pathogens have a significant environmental lifestyle. But what's really been missing is we often don't understand for these organisms what is the relationship between, the fungus in its native environment and what, ends up showing up in the clinic. And this is certainly the case through Aspergillus fumigatus, where historically a lot of the work is really focused on clinical isolates and understanding the pathophysiology of disease. And so at the end of our field study, I thought we really have the perfect data set to really answer this question, because we have a freezer full of environmental isolates. And so we took a subset of them and combined them with some clinical samples that were submitted to the German National Reference Center for Invasive Fungal Infection in Germany. And we performed whole genome sequencing on 252 of these isolates. And then because our samples were all from Germany, we also brought in some public data from publicly available data repositories to really make sure that whatever we found was also representative of the global Aspergillus fumigatus population. And this brings us to 300 genomes to answer this question of really, not only what is the genomic diversity of Aspergillus, which had not been explored, is what is the relationship between this fungus in the environment and the clinic. And I also like this image here that Sven Kratmet sent me. So this is 300, the number of genomes we were analyzed, spelled out in Aspergillus canidia. And so for our analyses, we opted to do de novo assembly and annotation. And this allowed us to take a really unbiased look at the genetic diversity, because we weren't constrained with genes that were only, say in our reference strain. And as just an example of what we can get from this, I've drawn 3 very simple genomes here. And we can compare them and see that, all 3 of them have this dark blue gene here. However, when we come over to here, we can see that genome 3 is missing this gene, as well as at this particular locus, there's some variation in what gene is present there. And an alternative way to think about this is that we can break genes into core genes, which are present in every single member of the species, as well as accessory genes. And these are things, that are not universally conserved across the species, and often these are implicated in things like adaptation to new environments. And then the pangenome is the word we give to this complete collection of core and accessory genes, and it represents the entire gene set for a given species. And so the first question we wanted to ask with our 300 isolates is really, what is the genomic diversity of this pathogen, and what is its pangenome like? And so this is a project that I worked very closely with Tongta, who is a PhD student in Jena. And what we can see is as the number of genomes that we analyze increases, so does the total number of genes, really highlighting that by only studying 1 or 2 strains, you're missing a lot of information. And the other thing that I think it's important to point out here is that you can also see that at the end here, we hit a plateau, signifying that when we look at these 300 genomes, we have a pretty good idea of the representative genetic diversity of the species. However, this sort of analysis doesn't really tell you much about the strain level, variation between. And so another way to look at this is what's called a presence-absence matrix. And so in total from our 300 genomes, we discovered just shy of 11,000 total genes. And those are all going to be laid out across the top here. And then our 300 genomes are going down. And if a gene is present in a genome, it'll be colored blue. And if it's black, the gene will be colored black. And what we found when we analyzed our 300 genomes is that only 69% of the total genes we found were core or present in all strains. An additional 9% of genes are what we call soft core. And so they're in most strains, more than 95%. But really strikingly, 22% of the total genes we discovered are much less conserved. And this amounted for any given isolate, 16 to 22% of its genome was not universally conserved among all members of the species. And so given that we found this large number of accessory genes, we wanted to take a closer look at them to see what else we could learn about them. And so one of the first things we notice is actually that these accessory proteins are shorter in length. And that's what we see here when we're measuring peptide length. And we can see that our core genes are the longest on average, and they are significantly longer than our soft core and our accessory genes. We also find that the accessory genes are much less likely to have an identified function. And one of the ways we looked at this is whether they contained at least one annotated interpro domain. And we can see that for our core genes, while more than 80% of them have at least one annotation, only about 50% of our accessory genes have them. So we as a community have a lot of interesting hypothetical genes to characterize in the future. And going along with that, I really wanted to highlight what having a pangenome adds over having just one single reference. And so one of the other things we did was kind of map our genes against the Aspergillus fumigatus chromosomes. And so this is the reference strain that many people in the field use. And actually what I want to do is highlight this thing down here, which is basically all of the genes that couldn't be mapped to anywhere in AF293, and really highlight that by looking at a pangenome, you basically have an additional chromosome worth of information that's really being ignored using a traditional kind of reference-only approach. And then the final piece of data that I wanted to show you about pangenomes is really to kind of put this in perspective. And so, I've told you that when we analyzed our genomes, we found that only 69% of its genes were conserved among all members of the species. And, is that a lot? Is that a little? And I think for fungi, it's actually in the middle. And so we can compare this to something like Nakasaomyces glabrata, which is also known as Canada glabrata. And we can see in some work from Tony Gabaldon's group that 94% of its genes are conserved among all members of the species. And then on the other hand, we have the pangenome for Zymoseptoria triticea, which is a very potent wheat pathogen. And some work from Daniel Kroll's group has really showed that only 59% of its genes are conserved among all members of the species from only 19 genomes. So I think the main takeaway is that there's really quite a wide variation of fungal pangenome structures, much like the wide variety in this wonderful kingdom we all study. Okay, and so we're interested in Aspergillus fumigatus because it's a human pathogen. And so one of the other questions we wanted to answer is really, how does this genomic diversity potentially impact virulence? And so the way that we address this is we compiled a database of 360 virulence-associated genes, and then looked for really high impact changes in these genes. And in about half of them, we don't find any high impact changes. But in 155 virulence-associated genes, we do. And that's what we're seeing here, where now we have our genomes going across the top, and then these 155 virulence-associated genes are going down on our y-axis. And if a gene is present and doesn't have any significant genetic changes to it, the square is black. And if you look at this heat map, you can see that there's a lot of black, indicating that even when we do identify these changes in virulence-associated genes, they're often in less than 15% of our strains. However, we also identify cases where, say, virulence-associated genes have been amplified, and there's now additional copies in the genome, or the reverse, where these genes have been either severely damaged by something like a frameshift, they've turned into pseudogenes, or they've been completely lost for the genomes. And so this is why we think, this genetic diversity is also relevant for infection. And so now I just want to bring you back to this question that I asked at the beginning, which is, what is the relationship between this fungus in its native environmental habitat and infection? And you could imagine two possible scenarios here. And so first is you have this really genetically diverse population in the environment. And when we're exposed to it, any one of these colors or strains is equally likely to cause disease. On the other hand, you can imagine a scenario where despite this really diverse population, maybe one lineage is more likely to cause disease, indicating it's more fit in human environments. And we find evidence for both, but we find strong evidence for this model here. And we'll go through that data now. And so the way we addressed this first was by building our isolates into a phylogeny, which is looking at the genetic and evolutionary relationship between them. And so you can see here that we used DAPC, which is kind of like a PCA for genomic data, to break our isolates into 7 distinct genetic clusters. And these colors at the end of the branches here indicate the genetic cluster that a particular strain belongs to. And then we can start to overlay additional data, such as, say, where in the world a strain came from. And here we can see that we don't see any relationships with, say, which genetic cluster a strain belongs in and where it was isolated. And we have, our strains from Germany mixing very closely with ones from the US and Asia. We can also start to overlay whether strains are from environmental origin or clinical. And here you can see, so we see these purple clinical strains that are distributed throughout the phylogeny, but we also identified a statistical enrichment for clinical isolates in one genetic cluster here, suggesting that these strains might be more fit in human infection niches. And so we want to take a closer look at the strains in the genomes of these strains. And here we notice something really interesting, and that's these strains have bigger genomes and they have more of these accessory genes than the other clusters. And so that's what we see here. And these accessory genes are the ones that not every member of the species has. And the cluster that's enriched for clinical isolates here in yellow has more of these accessory genes than many of the other clusters. And so given that I've told you they have more genes, you might want to know, well, what do they have more of? And so one of the ways we looked at that was quantifying the frequency of different gene ontology or GO terms in their genomes. And if we look at this heat map, we can actually see there's many different gene annotation so that have significant differences between the genetic clusters. However, you will also see that this one cluster here that was enriched for clinical isolates has this big red band that it has more of in its genomes than all the other clusters. And so what are these things? And we can see that these are things annotated as, being involved in oxidation reduction processes. This cluster has significantly more transmembrane transporters than other clusters, things involved in iron binding, as well as different metabolic processes. And so when we finished this study, we'd found really a previously unappreciated level of genetic diversity in our strains. And we had made some, pretty strong predictions that there would be functional differences based on their genomes, but we really didn't have a lot of data to back this up. And so now I'm really excited to show a recently published follow-up study that really kind of adds to this and gives some convincing evidence that these genomic differences also translate to functional differences. And so this is a project that was done in collaboration with the group of Yanni Panagiotou at the Leibniz HKI, which is also in Jena. And a lot of work was done by Mohammed, who's a PhD student, as well as Sasha, who's a postdoc. And what we did is we made a pan-genome scale metabolic model of Aspergillus fumigatus. And a metabolic model is a way to kind of predict in silico how a fungus or any organism will grow given, a set of media components, a set of metabolites. And it also allows you to do cool things like make in silico knockouts and see how, what the predicted biomass will be. And so now I'm going to simplify about 2 years of Mohammed's PhD into one sentence and say he made a pan-genome scale metabolic model. And then once you have this pan-genome scale metabolic model, you can then use this to basically take the genomes of all of your strains and map it back onto this and get an individual model for each one of your strains. And then after a bit of curation, you get a really high level collection of strain-specific metabolic models. And so a few minutes ago, I shared with you, that when we looked at the genomes of these isolates, we saw a really high level of presence-absence variation. And, only 69% of genes were shared among all members of the species. And so we were very interested to see, what this looks like at the metabolism level, because often in metabolism, you have quite a lot of redundancy for good reasons. But really strikingly, when we looked at the metabolic model, only 77% of the metabolic reactions that are in the pan model were conserved among all members of the species. And that's what we're seeing here. And so on the bottom here, we have our phylogeny, which is saying, what the genetic relationships are. And then on top of that, we have stacked their accessory reactions that not every strain has. And the color indicates kind of how common these are. And when we look at this, we can see that most of these accessory reactions, even though they're not in all strains, they're in more than 80%. But we can also see that there are some reactions that are much more rare, and they're in, such as these red ones, which are in less than 20% of the strains. And in data that I don't have time to go through today, we also did some experimental work to validate these predictions from the metabolic model. And so the last thing that I wanted to share you today was really, we built this cool tool, this collection of genome scale metabolic models that replicate the diversity of species. What can we use these for? And I want to share a very short vignette about what we learned from these about Aspergillus in the cystic fibrosis lung. And so this is a cohort of patients who have cystic fibrosis, and we had access to sputum samples before and after they became colonized with Aspergillus fumigatus. And we performed shotgun metagenomics on these. And what we found is that, after Aspergillus fumigatus colonization, the lung microbiome is significantly different. And that's what's indicated here in these pictures, where I won't go through the fine details. And then in addition to having an altered lung microbial community, we also see that accompanying this, there's an altered lung metabolome. And this altered lung metabolome really got us thinking that this is what exactly, a cool example to show what these metabolic models can use for, because really the input for them is a set of metabolites. And so what we did is we took the metabolome from these before lungs and the after lungs and compared how well Aspergillus fumigatus is predicted to grow in this collection of metabolic models. And here we notice that really interestingly, Aspergillus fumigatus is able to grow better in the media conditions and the metabolome conditions in the lung after Aspergillus fumigatus colonization, saying that really, in the cystic fibrosis lung, once they get colonized with Aspergillus fumigatus, it changes and restructures the lung microbiome, it changes the metabolic environment, and this facilitates the growth of Aspergillus fumigatus. And so I think I'm wrapping up now, and the thing I really wanted to highlight is that I think Aspergillus fumigatus is just an incredibly versatile pathogen, and to really understand it, I think one referenced gene and one genome is not enough. And I think what we've shown today with this, large degree of variation in kind of gene content as well as metabolic reactions really overlays well with an accumulating amount of evidence that there's also a corresponding amount of phenotypic diversity between isolates. And then in particular, I showed you that we think that this variation is relevant for infection because we see variation in virulence-associated genes, as well as in our data set, we identified an enrichment for clinical strains in one genetic lineage. And these genomes had unique features. They had larger genomes with more transmembrane transporters, iron genes. And in some data that I didn't have a chance to go to, as part of this metabolic modeling, we also could identify, using machine learning, some metabolic reactions that were also sufficient to differentiate clinical and environmental strains at the metabolic level. And so with that, I'd just like to acknowledge everybody involved in this. And so the field study that I told you about in the first part was done when I was a postdoc with Oliver Kurtzai, working in collaboration with the University of Halle. And then the pan-genome work was done when I was also a postdoc with Oliver, but working in close collaboration with some other groups in Jena. And then the metabolic modeling was really led by Yanni Panagiotou. And so, and I would also say that we have a PhD position in my group open. And so if this interests you, please get in touch. And with that, I'm looking forward to the discussion. Thank you.
Well, I think we've been treated to two fantastic talks, really amazing. So thanks to both. And just a reminder, for those who would like to ask questions to either Amelia or Jenny, do put those into the Q&A box, and Joe and I will take those forward. We've got quite a few questions already accumulated, and perhaps I'll just kick off with the first question for you, Jenny. And it's a question saying you've made all these mutants. I think that they're referring to the chitin synthase and CDA mutants and the accessory protein mutants. And the question is, what about susceptibility to echinocandins in those backgrounds?
Yes, we've looked at those, and there's you can see some variation and some increased sensitivity, but it's not dramatic, which was a little disappointing. We were hoping for that, but we are, as I mentioned, we are looking at sort of what the echinocandins are binding to. We've gotten some BODIPI and other fluorescently tagged echinocandins that we're able to look at, and Anna in the lab has started to look at the chitin synthase 3 mutant in terms of where the echinocandins are going, and she's seeing some very interesting results. So, but very preliminary at this point. So
great. Well, there's MycoTalks in the future.
Yes.
All right. Over to you, Joe.
Great. Terrific talk, Amelia. We have a number of questions that have come in, and to combine a couple of them, do you observe any correlation between differences in the core genome, accessory genome, and variations in chromosome organization? Or to put it another way, are the accessory genes in special regions of the genome?
Yes, and so I did not talk about that, but I think if you looked at the chromosome plot, there are a couple hotspots that are very, very different. As to why that is, I'm not sure. I mean, we're interested to look and see whether this is something that maybe is more, susceptible to crossover, as well as maybe more susceptible to something like a transposon jumping in, something about the sequence. But the answer is that yes, there are definitely some things. Aspergillus doesn't do quite as many of the cool, totally dispensable chromosomes like some of the other fungi do. For example, some fungi have these completely accessory chromosomes and they can completely get rid of one. Aspergillus doesn't do that. But yeah, there's lots of interesting things that I can't answer, but I would love to be able to in the future.
Terrific. Over to you, Neil.
Okay, so Jenny, in a one world context, there's a question here saying that they're noting that there are inhibitors of chitin deacetylases being used to control plant fungal infections. And is there any opportunities there to identify novel inhibitors for chitin deacetylases for treatment of cryptococcus?
Yes, and I think that's a great question and one that we've been very interested in. We have we've developed a screening technique that involves flow cytometry and looking at the brightness of the cells in flow cytometry, and we can actually see differences if we inhibit the chitosan production. We did a preliminary screen, and we did come up with compounds that reduced chitosan production. The one that we followed up on and looked at was really a signal transduction inhibitor. It was impacting the cyclic AMP pathway. But I think one of the things that we're interested in, and Anna in the lab has a lot of expertise in biochemistry and protein structure and that sort of thing. And we are very interested in sort of producing sort of purified chitin deacetylases and developing an activity assay for the Cryptococcus ones and actually doing a screen against those, as well as trying to identify structures and whether or not we can look at the, ideally we would be able to identify the structure and then maybe in silico try to figure out, of the whole complex, and then in silico try to figure out how we might design inhibitors for the Cryptococcus chitin deacetylases. The nice thing is these are outside the cell, so you don't even have to get them over the membrane.
Yeah.
So.
Great, thank you. Joe.
Okay, another question for Amelia. What kind of selective pressure do you think may be operating on those accessory genes in Aspergillus while they are in the environment? In other words, are the virulence genes accidental, or are they also under selection in the non-host environment?
Yeah, I mean, that's a question that I think about a lot, is kind of where these genes come from, what sort of selective pressures. So I didn't show the data, but in general, the accessory gene is under a much less stringent level of purifying selection. And so both the core and accessory are under overall levels of purifying selection, but the accessory gene is under a much less level of this. And I mean, I believe in this accidental kind of virulence hypothesis. And actually, I like to take it one step further and actually think that, the environment is kind of in a training school for fungus where they, through their interactions with other microbes, all the stresses they have, they learn everything. And so, I mean, my working model would be that through these environmental actions, there is something in the environment that is, causing these genes to, to have a benefit that overlaps with fitness in the host. But in terms of, who are the teachers in this environmental virulence school, we really don't know enough to answer that.
Great. Over to you, Neil.
Okay, so Jenny, on the more immunological side, it's a question asking you to speculate a bit more about the CDA induced in protection that occurs in the absence of CD4 cells at the point of infection. How does that work?
That's a great question. And I think this is, the area that we've been looking at, particularly Stu and Charlie up at UMass have been looking at, what are the PRRs, for example, that are involved in inducing the production? What are the, what other cell types are required for protection? They've been looking at complement and all kinds of different things. And so I think there's still a lot more that we need to understand before we can really speculate on why it is that the protection would continue in the absence of CD4 T cells. But I think that's that's the really important question, isn't it? So.
Thank you. Joe.
Amelia, there's a question about potential occupational risk for aspergillosis. For example, comparing composters and gardeners to those in agriculture or working in silos. Is there any data on this?
Yes, and so my understanding is, so yeah, there is, but actually they get a different kind of aspergillosis. And so it's really, in this case, kind of all about the host. And so if you're immunocompromised, you don't get the invasive form, but it's actually called farmer's lung. So people who work in agriculture, people who work, near composting facilities, they tend to actually get, rather than an invasive pneumonia, they get this really chronic inflammatory disease, kind of like an allergic bronchopulmonary disease. Which is, this is another great question, is how that might be different. So our strains, I think I forgot to say, were really from invasive disease from this one patient cohort, but it's really an open question of whether, this relationship between host and pathogen and, what sorts of different host environments selects for different fungal determinants.
Mm-hmm. I have a question that might cross-fertilize some discussion. And I found it very thought-provoking, Jenny, you're finding that if you don't have chitosan, the fungus seems to hyperactivate the immune system. And so I'm wondering, do you think of that as a sort of a cloaking mechanism? And might other human fungal pathogens also decorate their surface with chitosan, like Aspergillus fumigatus, for example?
Yeah, that's obviously one of the things that we've been thinking about, is whether or not this is really a way of masking the chitosan. One of the functions might be to mask some of the other epitopes that are in the wall. And, and one of the things that, this is also very preliminary, Joe in the lab has been looking at, the localization of chitosan in the wall and starting to see maybe it's not in the inner wall the way chitin has been proposed to be, but it may be more towards the exterior of the wall. That's extraordinarily preliminary data. So don't take that away, but that's definitely one of the things that we're looking at. And then I think as we start to think about sort of this, what happens when we grow the cells in unbuffered yeast nitrogen base, where you see where we're seeing this sort of pulling away of the wall, that's really the hyperinflammatory response, is because you've now unmasked all of these epitopes.
Great.
Actually, can I ask Jenny a quick question related to that?
Sure.
So I'm interested. I've never really thought about chitosan and Aspergillus fumigatus, and so you showed some data that Saccharomyces has a very different level or this ratio than say crypto. I mean, what is the story in filamentous fungi? What is the ratio like? What is the total amount like?
Yeah, and so the mucors, they have far more chitosan than Cryptococcus does. They have quite a lot. Aspergillus, there's only been a little bit of work looking at that. I think it was done by Jean-Paul Laggi, and he only found a small amount. So I don't know if it's as important a mechanism in Aspergillus, but at the same time, that was in one strain, right? And I think one of the things that your work highlights is that you actually have to look at lots of different strains. And actually, that was one of the things I had a question for you, which is you showed that different species have different ratios of your core proteins versus your accessory proteins. And I wondered if that was a reflection of their different lifestyles. So perhaps a species that has to survive in many different niches with many different, food sources. Does that have more of the accessory proteins? Does that increase the ratio of accessory proteins versus a species like if you looked at Candida albicans, for example, that's sort of an obligate, lives on the skin of people, and it's very obligate that way. So it has a much narrower environmental range. Would you see differences because of that?
Yeah, and I think that's a really good point, and you do. And so, I think so. Like Aspergillus has to live in all these different habitats. The environment can be quite changing. I think this is why. And I guess I would add that so Zymoseptoria, this plant pathogen, it's entered into, I think, what they call a devil's bargain, kind of where it wants to really overcome the plant immune system, and these evolves very quickly. And so it's basically given control over to transposons and let that generate extra genetic diversity at the cost of also some negative consequences.
Right. And so when you start to think about your clusters, is it possible you're seeing horizontal transmission between species?
We would love to answer that. And so this is Marion in my lab has started to look at that, because one of the things that kind of precluded that in our analysis is all of our work was done with short read assemblies. And so one of the key things you look for in horizontal gene transfer is synteny, and we really wanted better synteny than we had. And so we've gone back and generated kind of near chromosome level assemblies from a panel of 15 isolates. And we're starting to now be able to look more not only at these chromosome dynamics, but then also things like horizontal gene transfer, because I would really love to know where these accessory genes come from.
Joe, our job's turning out to be really quite easy here because we're just sort of getting questions coming from all angles. But continuing one of the sort of trains of thought there was a question for Jenny, which was looking beyond Cryptococcus and trying to apply the idea that chitosan deficiency might be a useful way of controlling maybe some things like horrific diseases is the context of the question, such as zygomycosis. So can this perhaps help us deal with those really awful diseases in the back of, say, coronavirus infections?
Yeah, I actually think that's a great question. And I think that there is definitely some possibility there. We haven't moved into those types of fungi yet because the gene complexity in chitosan biosynthesis in those particular species is enormous. I can't remember the number. I think it was like 17 chitin deacetylases, which if you want to start dissecting this from a genetic perspective is rather daunting. However, it does also sort of give hope that if we can figure this out in Cryptococcus, and if there are some potential drugs that we could develop that are chitin deacetylase or chitin synthase inhibitors, that perhaps those could be then extended to some of the other black molds and other species that have lots of chitosan. So.
And in the spirit of cross-chairing, I wonder if Joe would mind if I ask Amelia a question, which is, given the amount of genetic variation you see, Amelia, between isolates of a species, do you think we're jumping in too quickly to propose cryptic species, and that actually it's really reflecting the variability you're seeing within a greater population, and actually adding a new name is helping us or hindering us?
Oh man, I mean, I think that's taxonomy is such a complicated issue. I mean, so my understanding is, I mean, I guess what do you call a species? But often in many cases, it's kind of where the boundaries of mating lie. And so for Aspergillus, I'm pretty confident that what we have is really Aspergillus fumigatus sensu stricto. But there are some cases, you know, maybe in crypto as well as, you know, Fusarium, where these boundaries are much more fragile. Yeah.
And some of them don't mate, so I guess we have
Yeah,
to look at
well,
the natural
well,
variation and make decisions.
I guess I would say we don't have evidence that they mate, not that they don't mate.
Oh yeah, don't mate yet, right? Okay, back
Amelia,
to you, Joe.
there was a question that came in along these lines, asking whether or not sexual reproduction is occurring in, say, in compost in the environment. And do you have a sense from looking at all of your isolates genomes how recombining that population might be?
Yes, I guess to answer the second part, we don't have an idea, but we'd like to. And so I think with longer read assemblies, we'll have a lot easier time sorting out these crossover events. But I mean, to answer your first question, I think for a long time we didn't really think Aspergillus was so sexual in nature, but I think some evidence that's coming out of the Netherlands in the group of Evelyn Snelders and Ben has really shown that. Actually, I'm not sure if it's published, but I I believe they can find some evidence of these sexual structures in compost.
Terrific.
So Jenny, I'd like to combine one of my questions with one of the questions, which is getting into the biochemistry of these enzymes like the CDAs and how stable they are. And I really liked your model about how the CDA was tethered close to the membrane, and that was necessary to actually deacetylate the chitin. And I wondered whether what you were saying was that actually the deacetylation is required to occur before the antifibril cross-linking hydrogen bonding of the chitin fibril takes place. And once that's happened, it's rather inert to deacetylation. Is that the kind of way that you see it? So it has to be in that kind of open nascent form to be accessible to modification by deacetylation.
Yeah, I think that's quite possible. And you know, with the CDA4, which is the one that sits out in the membrane, the substrates that they used were short molecules, so they weren't in that sort of crystalline stable form. So you know, and so we haven't been able to show. I don't know that anybody's been able to show, you know, if you take chitin out of a Cryptococcus cell wall and treat it with deacetylases, whether or not you'd see any deacetylation at that point. But it does seem to be that once it is formed, it's pretty inert. So
a prediction might be that if you treated the cell with chalcofluor white, which binds and makes the nascent chitin less accessible, that might reduce the capacity of the deacetylation to occur.
Reduce it or increase it?
Well, actually, yeah, I guess you could say it might keep it in the open form, or it might actually be a sort of steric blocker to.
Right.
Yeah, so maybe that's not a great experiment because you can use both alternative mechanisms to explain.
Right, but if it did something, that would be interesting, right? And then
right.
Yeah. But yeah.
Joe.
I had a question for Amelia about your studies in the field, and you found a limited number of isolates that were becoming azole resistant, but did you have a chance to sequence them and look at what the mechanism might be? And were there any mutations related to the ones that people have found elsewhere?
Yeah, and so actually, we always tell science in this beautiful story, but actually the reason we sequenced all of these genomes to begin with is we were actually quite interested to use genomics to look for novel resistance mechanisms. So we did sequence some of them. The answer is we didn't really find a lot. And I had hoped that we could use kind of these pangenomes to look for maybe novel genes associated. And we did some kind of basically statistical testing to see if there were genes that were specifically, more associated with being in a resistant strain. And we didn't find anything super convincing. I guess I wouldn't say that it's not there, but at least the hits we got back were largely unannotated. And so we didn't really take a stab at experimentally characterizing them. But since you brought up resistance, and I know you like epimutation, I can also share a little tidbit, which is that I didn't show the data, but when we have a couple fields that we were able to monitor, year in, year out over multiple years. And here we notice that actually the susceptibility, is low, and then it goes up in the after period. But then when we come back to that same field, it's kind of reset, indicating that whatever resistance is there, it may not be genetically encoded.
Okay.
Yeah, I was going to ask whether or not you thought there might be any aneuploidy, like the heteroresistance in cryptic.
Yeah, so we have looked for that in our resistance strains, and we don't see evidence for that. But I think that might be because Aspergillus is, Canada it's really famous for, but it's a diploid organism, and so it's just a little bit easier, I think, to generate and carry these aneuploidies. We don't see this, but it doesn't mean that, it isn't something that contributes. And I think in Aspergillus flavus, it was recently published in Experimental Evolution that they were able to see a little bit of that.
Okay. Now, I'm going to have to apologize first to our speakers, but also to some really good questioners. And we've grabbed the questions and we will send them to you, but we will actually run out of time on the meter. And so you can have a chance to respond to those directly to the people who asked them. There are some really good questions, and I think actually it's likely that it could continue coming. But can we just thank both Jenny and Amelia for wonderful talks, and do encourage you all to join us again for that next set of talks, which will be on the same channel in a couple of weeks' time in November, and do come along for that as well. Thank you very much, everybody.
Thank you.
Bye.